Welcome to Signals Synapses, the podcast of the Max Planck Institute for Biological Cybernetics in Tübingen, Germany. Today, we'll take a deep dive into cognitive neuroscience. I'm happy to welcome Romy Lorenz, leader of the Cognitive Neuroscience and Neurotechnology Research Group here at the MPI. Thanks for joining us, Romy.
Hi, thanks for inviting me.
Romy has a multidisciplinary background encompassing psychology and human-machine interaction. In her current research, she wants to understand how complex mental processes such as thinking, problem-solving, and decision-making are supported by structures in the brain. We're also joined by Christina Bruckmann, who is a PhD student in cognitive neuroscience here at the MPI. She studies how perception interacts with our ability to predict timing and is particularly fascinated by the phenomenon of consciousness. Welcome to this episode, Christina.
Hi, happy to be here.
We're going to take a deep dive into cognitive neuroscience today. What is it and how did it originate? What are neural networks in the brain and how can we explore them? And how might stroke patients benefit from fundamental research? To kick us off, Romy, can you tell us what cognitive neuroscience is actually all about?
Yeah, that's a good question. So maybe we can go back a little bit and think about where the discipline emerges from, which is psychology. And psychology actually has really ancient roots. I mean, already like really ancient philosophers like Aristotle and Plato were interested in questions that concern the psyche, like how does matter give rise to the mind? And in the beginning, or like quite for a long time, we had very rudimentary ways to look at the psyche. So we looked at really indirect measures such as observations, behavior, reaction times, and these kind of things. But then as technology evolved, we now then could look into the brain using brain technology, such as electroencephalography. This is something Christina works with, right? Or functional magnetic resonance imaging, something that I work with. And once we had these technologies, also like the discipline of cognitive neuroscience emerged, where we could use these technologies to look into the brain to study how the psyche works.
In the study of cognitive neuroscience, there's a lot of different ways, or let's say philosophies, of how we can look at the brain. So some people just study one specific region. I think that was very popular in the past, and other people focus on a very specific function. But your work focuses much more on networks, so different regions that interact at the same time to accomplish a task. What are some of the challenges that come with that approach?
Yeah, so it's actually interesting that you're bringing this up because there are these different ways on how obviously like you can approach cognitive neuroscience, and there is like the very popular outside-in approach that you mentioned, like people focusing on like a specific cognitive process, such as like working memory, which means that you hold a thought in your mind for a short period of time. And then they study what kind of regions are involved with that. And I'm tending more into like an inside-out approach, basically trying to first focus on a structure and then trying to understand its role in human cognition. And the challenge with that comes from the fact first, like how do you define these structures? There are different ways of doing that. Like obviously anatomy is like one way of doing that. But we also know that especially in the regions that are involved with intelligent behavior and behavior that I'm really interested in, that function and anatomy not always completely align one-to-one in all different humans or individuals. So there really is the challenge on how do you even define like the structure to begin with.
Before we go more into the depth of the difficulties, can you tell us a bit more, like what do you mean by intelligent behavior? Can you give us some examples?
Yeah, so I'm interested in intelligent behavior, but I'm also very interested in the structure, as I mentioned, that is kind of often involved with intelligent behavior, and this is called the frontoparietal network. So this is a network a network consisting of like frontal regions in the brain behind your forehead and parietal regions, which is like more to the back and the top of your brain. And the frontoparietal network is involved in many everyday activities. So just think about like a vacation you're going to, you're renting in a car there, and you're going onto like into a new city that you don't know of. You need to follow the GPS. You need to kind of understand like the different traffic signs and different languages. You may need to talk to your partner who's also trying to navigate you. And like just really juggling all these different tasks and switching between all these different mental demands, the frontoparietal network is involved in. Another one is like if you go to the grocery store, you have like, you try to remember your grocery list. You're trying to search in the aisle for like your favorite cereal brand. Like you have this kind of selective attention. You're trying to not listen to like this annoying music or so. You're trying to avoid distractions. This is again all the frontoparietal network helping you with like these kind of mentally effortful tasks.
So what is actually a network in the brain?
A network in the brain really just means that brain regions never work in isolation. But you can think of it as like an orchestra where the different brain regions are the different instrument sections like violins, percussions, drums or so. And they really need to work in synchronization to that intelligent behavior emerges from that.
Especially now that you say there are so many different functions that you need to carry out while the network is involved. I'm always wondering for these network approaches, how can you define that these regions actually interact and form a coherent network rather than just being active at the same time because they commonly are involved in similar situations, or how you distinguish that also from other networks that might be interacting?
Yeah, so again, this very much depends on the method you're using. So for example, in like a fast temporal method such as like EEG, you have a bit of better ways to seeing how, for example, regions are talking to each other because you can really resolve it in time much faster. And with a technique that I'm using, functional magnetic resonance imaging, like a problem really is with like the more conventional way of doing it, that we can't quite tell the difference whether they're just co-activated at the same time or whether they really kind of influence each other. But this is also like an interest of the research I'm doing is to actually zoom more into the brain, not just looking at like a single region and how these different regions kind of communicate with each other or co-activate with each other, but I'm trying to really zoom in into the different layers of the brain. So you can imagine the brain being like an onion, having different layers. And these different layers actually receive, some of them receive input, some of them basically project back output, and some of them are kind of processing hubs. And then by focusing on these layers, I can make more inferences on really how these different regions within a network are connected and whether there is really like basically some kind of, we would say causal information flow, meaning that there's really like one region influencing the other.
Okay, so that means that you think beyond what we see with just normal fMRI, there's more information if we look closer at the different layers and rather than just at the specific network and the full activation there, right?
Exactly, yeah. Okay. So my hope is like with this kind of more zoomed in approach where I'm still using fMRI, but like almost like under a microscope. Like I'm really zooming in with unprecedented detail. And only like a few, like there are not that many scanners in the world, like MR scanners, that allow you to do that. Like you need a really high magnetic field strength. And my hope really is like with this that we can move beyond like these more descriptive mappings that we currently have or like more descriptive descriptions of network activity and try to better understand really how do these regions interact. Like what is the precise timing? What region is influencing what other region? Because that is currently actually not really easy with any standard methodology we have in humans. In animals, we can already do that, but obviously there are like other challenges with studying intelligent behavior in animals.
You mentioned before that the frontoparietal network is involved in a lot of different functions, and you gave examples from daily life. But I guess when you're studying the network in the lab, you cannot have people go shopping, for example. So how do you get the network to activate once people are in the lab?
Yeah, I mean, that's like very much a challenge in our field in general, that we have to be reductionists. So we really have to break down really complex everyday life scenarios to very simple tasks. And I think often it's much less glamorous than probably people think in the outside what we're doing. So for us, it's really like inviting people into a scanner, meaning they have to lie down, and then we give them often like a screen that they can look at, and then they just have to do some very simple tasks. So you can imagine, for example, when we want to study a concept such as working memory, we call it, meaning that you want to hold like a thought in your mind for a short period of time. Let's say like a one-time password you need to remember for a little bit. And then we just really give people like, for example, images or like words to remember for like a short delay period of like 13 seconds. And then we probe them again whether they remember that or not. And so, yeah, as we also have to make our experiments very clean in a certain way that we can really then understand that the concept we're interested in is not kind of confounded by any other variables. So we have to do very simple experiments. But I mean, there are also movements in the field that people do like VR-based experiments. So people on roller coasters trying to induce like more complex emotions, or gaming is also like popular, or also even movie watching compared to just like people lying at rest. But for my research, because I'm focusing on like these more intelligent tasks, people are still kind of involved because it's not too boring. Like they actually do have to quite engage to be correct. So that's also something we always need to kind of watch out for, that the tasks are difficult enough for people to engage their frontal-parietal network, but not too hard that they get frustrated or that they just completely fail.
That also leads me to something that I wanted to ask you about networks specifically is you mentioned the individuality of people and participants because we're studying humans and not specific physical phenomena, I guess. So are all of these networks and how they activate very coherent across people? So once you know 10, you know all of them, or how does it work?
Yeah, so this is also like a very interesting new area that is evolving in the field. So historically, we would kind of do these group approaches, meaning that we assume once we average brain activity or brain network activity over many different people, that this will give us like kind of insight into how these networks act or interact in general across the whole population. But we also now know that many of especially these higher level networks, such as the frontoparietal network that's engaged in very intelligent behavior, actually has the highest intersubject variability. So this means that actually it's really idiosyncratic across different subjects or different people that we're measuring. Even like healthy young adults can have very different configurations of this network. And so we're interested in really looking at individual features of these networks and trying to avoid like doing any group averaging that really gets rid of these fine-grained details.
What do you think is the source of this individuality in the networks? Because a lot of simple like systems that are more basic, like vision, tend to be very uniform across participants. So why would these networks be so individual?
Yeah, I think that's a great question. And as you mentioned, like certain networks, visual network, motor network, they're actually quite similar and across subjects. But then these higher level networks are very unique. And I think like the current understanding is that it's also networks that actually evolve quite late in life. So for example, the prefrontal cortex, which is part of the frontoparietal network, also part of other networks, but evolves quite late. Like it matures like during your adolescence. And there can be also quite a lot of genetic factors that may play a role that kind of change the kind of development trajectory of these networks. So it's probably like an influence of obviously like your genetics you have, but also the life experiences you have, that these networks can look very different.
I know that this is a question that usually fundamentally researchers are not that fond of, but I'm very curious, especially now that you personalize it a bit and apply it to the individual. Do you see any applications in the near or far future for this research?
I mean, we always have to think a little bit of applications, even though I have to say I'm quite a basic scientist and I'm also really interested in basic science. But some parts of my lab also do neurotechnology. So obviously there is like also a link there that you could at some point employ it like in different ways, even though I have to say I also use neurotechnology again in a different way than maybe usually people talk about it. I really use it as an experimental paradigm that helps me to answer questions that I couldn't answer before, but still related to basic neuroscience questions. In terms of how we apply it, I mean, there's always the link to brain disruptions, so clinical and neurological disorders. And especially with my work on the frontoparietal network, this is a network that is really involved in many of these disruptions and in many, like it shows disruptions in many clinical and neurological conditions. So I do think there can be quite beautiful links then trying to also understand how we could advance like therapies or even just really try to understand like a disease mechanism.
What kind of diseases are we talking about there?
It's actually quite a lot, but like there is, for example, ADHD that is linked to the frontoparietal network, and that there are disruptions with the communication of this network to reward centers. Schizophrenia has shown links. Again, like disruptions on how the frontoparietal network communicates with other networks, and also depression. That for depression, it's very typical that people get like kind of stuck in certain thought loops. So, and the frontoparietal network is a network that kind of is a flexible one that aims to try to switch between different states. So there's also like a link to depression here.
So I have a hard time wrapping my mind around how you would actually make an intervention for, say, a schizophrenic or a depressed person. Like, where does neurotechnology come in there?
Yeah, so, I mean, again, like I think the way I use neurotechnology is very different than probably other people maybe know of neurotechnology. I think a lot of people now probably think of Elon Musk's Neuralink. And this is, for example, like an invasive neurotechnology where, I mean, they're currently trying to record and then also manipulate like brain activity with that. I think, I mean, for me now, when I talk about like trying to understand the frontoparietal network, I'm thinking more about like first trying to really understand what is the mechanism of this disease, because this is actually really challenging. Like probably also, as you know, even though brain science is not like such a young discipline anymore, especially with like mental disease, we actually haven't made like immense progress in understanding it or even therapies. So I think it's first really trying to understand a disease mechanism before we can then talk about therapy. But like I think some neurotechnology that could be thought of as a therapeutic might be non-invasive brain stimulation techniques, or also, I mean, invasive brain stimulation techniques are also therapies, which actually really successful for Parkinson, where we have deep brain stimulation that actually shows like really remarkable improvements and for the quality of patients' lives.
You mentioned before that you, in your research, use neurotechnology slightly differently than is usually assumed. Can you explain how it helps you, what you do with it for your research?
Yeah. So again, like the neurotechnology I use, I can also refer to as like brain-computer interfaces. And as we talked about it, like normally brain-computer interfaces are also used more clinically, for example, or that, for example, people associate this with people having maybe EEG activity being recorded from their brain of patients that are, for example, paralyzed, and then they maybe can control a wheelchair with certain commands they send via their brain. And I used to do, like I had actually like during my master thesis and these things, I worked with patients on this side of the brain-computer interface spectrum. But then during my PhD, I actually really moved trying to use the same technology in a way, meaning that we do record and analyze brain activity in real time, but then use it as an experimental paradigm. So meaning that this also allows me to quickly adapt experimental tasks in real time based on a participant's brain activity. And this gives me the chance to actually do many more small experiments than you could standardly do, because you can efficiently search through this kind of hypothetical task space or experimental task space more intelligently than you could do without analyzing the brain activity in real time.
So that helps you then with the very strong individual differences you said that you find in people, so you can personalize the experiment, basically, if I understand
Exactly.
correctly.
Yeah, exactly.
Okay.
We can really look at, for example, what kind of different sets of tasks engage the frontoparietal network in one individual over the other. And this is, again, is also something interesting because we have done this also in stroke patients where we really showed that this really gives very different results for different patients with stroke. And this is also kind of logical in a way because no stroke is the same. But again, you have to think of how normally the field is doing that. Normally the field is testing a cohort of patients and they all get administered the same kind of cognitive tasks. But then we show actually that this is not like the best approach because they show disruptions in different cognitive processes.
So while also being useful for a research setting, you think also this methodology in itself would have clinical applications and advantages?
Yeah, exactly. So that was like the hope that we could showcase this with this one study we had. We first showed this, as mentioned, in also healthy people to really show there is like individual differences between healthies, but it's even more remarkable, obviously, in stroke patients. And then you could think once you identified like these individual functional disruptions in stroke patients, you could then take this also and have people maybe rehabilitate on those tasks or on these cognitive processes or combine it again with brain stimulation or so. Yeah, it's still very much like needs to be researched and it's very exploratory, but it's definitely like an avenue that is interesting to pursue.
So when you mentioned before that you use neurotechnology also to design the tasks, one thing I was wondering about is that if I understand correctly, the algorithm afterwards tells you which tasks activate the network in a specific individual, but does it tell you why? Can you deduce why in that specific person this task is active? Can you find like a common denominator across tasks maybe?
Yeah, that's a good question. So the algorithm can't say me why. Like the algorithm is really like it's you can really think like it's not a simple machine learning algorithm, but it's like a machine learning algorithm, and it uses like it's within the Bayesian framework, so it uses certain priors, but the priors I give in. So meaning.
Well, sorry, I'm just gonna jump in here. What do you mean by all that?
So this means that my algorithm isn't completely naive to the question that I pose it to answer. It means that I will give some prior information that it will leverage. And this information, for example, is that let's imagine like a space. Like let's imagine like we have a 2x2 grid, and this is filled with different cognitive tasks. And I will tell the algorithm the tasks within this 2x2 grid, when they're close to each other, they should elicit similar brain responses. And this is like information that I will use from prior experimentation or even from meta-analysis or so. And this is a prior information that the algorithm will use. So, and that's also then how it searches through that space. And so when for example, the algorithm will move to another task because, for example, like a previous task was suboptimal in activating the frontoparietal network, for example, it will do so because of the data that it got fed in. So it won't tell me why it moved there because it's kind of logical from just the computations it's done it will just try, like it's an optimization approach. So it's called neuroadaptive Bayesian optimization. So it's just trying to optimize an unknown function. And when it basically moves to another point in that 2x2 grid, this is because like it was suboptimal before.
If I'm understanding this correctly, what you're saying is instead of just having your algorithm run a blind search looking for what's actually right, you just give it information that it already can work with so that it can do an informed search. Right?
Yeah, that's correct. So it's basically not a random search and not an exhaustive search, but by using this kind of information that I fed it in, it can do a much more intelligent and efficient search.
And then afterwards, do you analyze which tasks have been selected and what the common factors are among them to deduce something? Or is your prime goal to just activate the network, just to activate it?
Yes, in my previous research, when we first introduced this kind of method, the goal was to understand what kind of tasks basically activate like a particular part of the frontal parietal network versus another part of the frontal parietal network. So we kind of tried to dissociate that. And that was also already quite exciting. But the challenge really is to really then understand, as you say, what is the common denominator among those tasks. And this is something where I think you would need to add like computational modeling or so as a next step onto it, because it's actually really hard to understand, like especially with abstract functions as we're working with, what is like now the underlying factor and what is like really the function that unites it all in a way.
From what you've just said now, this sounds like really, really interdisciplinary. So I imagine that in your group you work with people who have a background in machine learning, maybe psychology, maybe neuroscience. Is that so?
Yeah, so I would say cognitive neuroscience in general already is quite interdisciplinary. So the training in itself is already quite interdisciplinary. In my team, I do have people who have like also done a bit mathematics. People are also interested in physics, but mostly I'm still recruiting a lot from cognitive neuroscience because I also noticed it's actually easier, for example, to teach cognitive neuroscience how to code and how to think to think more mathematically than vice versa. It's actually really difficult for like sometimes an engineer or like a mathematician to really teach them how to design an experiment or how to really think of a research question. People can do that, but I, from my personal experience, found it the other way easier, and it's also like the way that I came into the field. So,
how did you originally become interested in cognitive neuroscience? How did you start out?
Yeah, so I think I have quite a unusual trajectory, I would say, because I started with studying business psychology when I was 18, not really knowing what it was, I guess. And within business psychology, the thing that really fascinated me was kind of cognitive ergonomics, we called it. So it's how do we adapt machines to users? That was kind of the avenue that I was focusing on. And I was doing an internship at Daimler in their customer research center, where we looked at driving assistance systems. But then I also felt, I really dislike cars and car companies. So I started thinking, I should go more into like the medical field, because I also know you actually can also do this with medical assistance systems. So I started doing internships also in research institutes that focused on like surgery or assistance systems in surgery. And then I started my master's because of this kind of interest in human factors, which is again human-machine interaction. And there I was the first time, there I encountered brain-computer interfaces for the first time. And I had like just amazing professors there who just really sparked this interest in me. And so then I started like taking my first kind of neuroscience classes and that's when I got really hooked by the brain, I have to say. And I think it really kind of also, I always felt everything what I was doing before, like these behavioral analysis and so on, were really indirect. And then I could really look into the brain. And then once I encountered EEG, I was like, but it's still like so limited. And I really wanted to look like more closer into the brain. And that's when I really wanted to do functional magnetic resonance imaging. So I applied for a PhD then in that area. And that's how I ended up in the kind of cognitive neuroscience, neurotechnology field. I really came from the brain-computer interface field, and I knew I wanted to do something with cognitive neuroscience and machine learning.
And that's all the time we have today. A big thank you to Romy Lorenz and Christina Bruckmann for the fantastic insights and thoughtful contributions. And of course, thanks to all of you, our listeners, for joining us. As usual, you'll find links with further information in the show notes. If you enjoyed today's episode, don't forget to subscribe and share with your friends. Until next time, stay curious and keep listening.